ATP synthase is an integral membrane enzyme that catalyzes the synthesis of ATP from ADP and inorganic phosphate. More specifically, this enzyme uses the proton electrochemical gradient produced from the electron transport chain to allow protons to flow down their concentration gradient and power the phosphorylation of ADP. This scene represents the inactive ATP synthase. The F0 subunit is contained in the inner mitochondrial membrane and consists of a C ring, which is alternating orange and yellow helices, and an A subunit colored in green. This C ring allows protons from the inner membrane space to flow into the mitochondrial matrix. More specifically, a proton from the inner membrane space can enter a half channel, then migrate around the C ring, and finally enter the matrix through the other half channel. The A subunit plays an important connecting role, joining the brown B subunit to the C ring. Moving up in the structure, the F1 subunit is contained in the mitochondrial matrix and contains the following structures. First, the epsilon subunit is colored in dark gray. Next, the gamma subunit is colored in red. The top subunit from this view represents the delta subunit shown in magenta. These three components make up the central stalk and are responsible for rotating the enzyme. The helices of the gamma subunit extend into the alternating alpha and beta subunit shown in cyan and blue, respectively. These subunits include the catalytic sites where ATP synthesis occurs. Lastly, the B subunit, or the peripheral stalk, is colored brown and connects the F0 subunit to the F1 subunit. Zooming in, we can see an active site of ATP synthase. In its inactive state, ATP synthase contains a bound ADP molecule that is colored by element. The adenine of ADP participates in pi stacking with an active site tyrosine residue represented in magenta. The threonine and lysine residues represented in yellow participate in stabilizing hydrogen bonds with the ADP phosphate groups. Additionally, a magnesium 2+ ion, shown in green, forms a stabilizing interaction with the negative phosphate groups of ADP. This charge stabilization allows for the negative charges of the phosphates of both inorganic phosphate and ADP to more easily approach each other to form a phosphodiester bond.
The enzyme must undergo a complex conformational change to perform ATP synthesis. First, the C ring rotates the central stalk. This allows the gamma subunit to rotate and initiate conformational changes in the beta subunits to aid in ATP synthesis. There are three conformational states of the alpha-3-beta-3 hexamer that each alpha-beta unit will change between. These include the open, loose, and tight forms. The open state allows ADP and an inorganic phosphate to enter, and at the end of the synthesis, open conformation releases ATP. The gamma subunit will rotate 120 degrees, changing the conformation from an open conformation to a loose conformation. In the loose state, the ADP and inorganic phosphate are both stabilized by the magnesium ion and the hydrogen bonds of the active state site residues to hold the substrates in position for the reaction. Next, another 120-degree rotation of the gamma subunit changes the loose conformation to a tight conformation. In the tight state, the ADP and inorganic phosphate are brought close together to form a phosphodiester bond and synthesize the product ATP. The gamma subunit will undergo one more 120-degree rotation to return the beta subunit back to the open conformation to release the ATP. Overall, the three beta subunits undergo this conformational change, each in a different state at one time. From this view, we can see the difference between the active site residues when ADP is bound compared to when ATP is bound. The residues in the ADP bound state are colored in cyan, and the residues in the ATP bound state are colored in yellow. ATP synthase changes from its ADP bound conformation into its ATP bound form through rotation of the alpha and beta subunits of F1.
Here, the light cyan helices represent the alpha subunit and the darker blue helices represent the beta subunit. The multicyclic molecule present in the active site represents ATP. the negative oxygen atoms on the 3 phosphate groups are shielded and thus stabilized by the positive magnesium ion. Additionally, there are several hydrogen bonds holding the ATP into the active site. The 3 notable bonding residues, serine, asparagine, and tyrosine, are represented in yellow. These polar residues help stabilize the polar ATP.
These two scenes show the protein from a top-down perspective. That is, we are looking down onto the top of the F1 subunit. From this angle, we can further see the important conformational changes that occur as the protein rotates. As the structure flips between scenes, we can see the rotation of the alpha and beta subunits along the B stalk as it rotates during the first conformational state. This last scene compares the enzyme in the first and second conformational states. Most notably, the central stalk shows an obvious rotation with the epsilon subunit moving from behind the gamma subunit to in front of it when flipping between the first and second conformational state scenes. Some rotation can also be seen in the C ring, and there's a slight change in shape in the alpha and beta subunits.